Xtalks is the industry leader in connecting life science, food, healthcare, and medical device professionals. The Xtalks Life Science podcast features insightful conversations with industry experts about all things pharma, biotech, and more.
Hello and welcome to the Xtalks Life Science Podcast. I'm Ayesha Rashid, Senior Life Science Journalist at Xtalks.com, and this week I'm delighted to introduce a very special guest on the show. We have with us today Dr. Marshall Fordyce, who is the founder and CEO of Vera Therapeutics, which is a company focused on developing treatments for immunological diseases that improve patients' lives. Dr. Fordyce has more than 15 years of experience leading teams in drug discovery, development, clinical translation, and commercialization of new treatments. Before founding Vera, Dr. Fordyce was the founder and CEO of gene editing company TrueCode Gene Repair, served as an entrepreneur in residence at Kleiner, Perkins, Caufield, and Byers, and as senior director of clinical research at Gilead Sciences, where he contributed to 7 new drug approvals. Dr. Fordyce currently serves on the board of directors of the Albert and Mary Lasker Foundation, and he received his bachelor's in arts from Harvard University and his MD from Harvard Medical School. To discuss new innovations in therapies for immunological diseases and the work that Dr. Fordyce is helping lead at Vera Therapeutics, let's turn to him now. Thank you so much for being on the show, Dr. Fordyce.
Thanks so much for the opportunity and your interest.
Thank you. And so I'd like to begin by asking you about your journey in pharma drug discovery in all of these leadership roles and what ultimately led you to Vera.
Well, it all begins with medicine. I was an undergrad who was interested in a career in medicine because I saw that would be a rewarding way to give back. So I began as an idealistic young medical student at a time when really there was a growing interest in doing something about global health. Many of us who were in college in the late 90s had seen the diseases HIV/AIDS, tuberculosis, and malaria become global problems, but our mentors were really active in doing something about it. And actually now is a really interesting time to talk about that, given currently disruptions in funding for example, for PEPFAR, which is U.S. aid to fund HIV treatment globally, including over 30 million people with HIV in sub-Saharan Africa. So that was really the beginning for me as an idealist who wanted to do something in the world and with my life. And I went to medical school with fantastic mentors throughout. I'm very fortunate to have mentors who led me to solid clinical training and ended up doing my internal medicine residency in New York. I was married at the time, and we moved from Boston to New York. And I was at NYU Bellevue Hospital, which was a fantastic training ground for a young physician. I stayed as a senior chief resident, so that was early experience as a leader in a complex medical system and in training. And then I kept with my interest to become a clinician to take care of HIV patients. And I got my fellowship in infectious disease with a great mentor named Scott Hammer at Columbia Presbyterian. At a certain point, I think a lot of physicians decide if they're going to take care of patients full time or turn to some other avenue that could improve what we offer patients or how we deliver care. That could be basic research, clinical research, software, whatever it is. But I think most physicians, at least in the US, hit a juncture where they know that we can serve patients better than what we're currently doing. And I was certainly one of those people, certainly interested in HIV and what we could do in terms of improving the medicines that we offer patients. And that was my step into industry. So after my fellowship, I did about 2 years in a translational medicine program at Rockefeller University, Aaron Diamond Age Research Center, and worked on a drug development program in the academic setting. Went to my first IND meeting as a young trainee, and I thought, wow, this is really how you make a medicine. And that was, I can't remember how many years ago it was, but it was, just early in my training. I started my family, and I started to get serious about, if I'm not going to take care of patients full time, if I'm not going to pursue grants to continue clinical research, maybe I should think about a career in industry. And I got very lucky. I had the opportunity to move out here to California. So from New York to California back in 2010. So that's now been 15 years. And had a chance to join Gilead Sciences, which was the leading company in developing new medicines for antiviral drugs for infectious diseases. And that, of course, was established in HIV. So I joined a well-oiled machine in HIV drug development, also hepatitis B. And the year I joined was when Gilead really took a step into hepatitis C, which, of course, was curative treatment and an incredibly exciting time. So from 2010 to 2016, was able to learn about drug development coming into industry. I think you appreciate very quickly that many types of expertise need to come together as a team to develop drugs. I started in clinical research and grew to become a project leader. And that gave me a strong appreciation and understanding of all the different disciplines that are required. And then around 2016, I started to imagine three changes. One, did I want to work in an area outside of HIV? Because we had made so much progress in HIV treatment. So one was a change in therapeutic area. The other was a change in organizational size. So after you work for pharma or large biotech, you realize that at a smaller scale, you actually have more optionality and more nimbleness as a professional trying to create a new medicine. And I started to recognize that opportunity. And third, what was my role? And I was looking at chief medical officer positions, and I decided, if I got a chance to choose the science, build the team, tell the story, raise the money, establish the culture, I was ready for that. And that began the Vera story, which started in 2016 now. So, it's been about 8 years, 8 or 9 years. And I founded the company. We were called CDF Therapeutics after the first letters of my kids' names, because we didn't have any assets or any platform. But within about 6 months, I was in front of the Kleiner Perkins investment committee, pitching an idea to develop a non-CRISPR gene editing technology company that we called TrueCode, and capitalized the company to about $30 million between Kleiner Perkins and Google Ventures. And we're on our way to our first company building experience. So that's how it all began. That was a mouthful, Ayesha, but that brings us up to kind of beginning the company.
That was fantastic. No, thank you so much, because it's so important to really set the stage in terms of how things evolve and come about. And so it's always fascinating to hear about those kinds of journeys. And so thank you so much for outlining that and really going from academia to large pharma and then to, a startup, a smaller biotech. And like you said, the flexibility and nimbleness that affords you. I think that's so wonderful to hear about. And what I'd like to hear more about now is really in terms of Vera and its core missions and goals, and what is it attempting to achieve?
Yeah, Vera came out of TrueCode. So when we began TrueCode, we found within a short amount of time that the technology, which was preclinical at the time, was not ready for further development commercialization. We had to pivot in about 2019. And we had this incredible opportunity because we had a team, we had money, we had committed investors and a board of directors that was supportive of the company pivoting. And we were doing active business development. And we asked a general question. We thought, you know, there are about 1 in 10 people on the planet and in the United States who have an autoimmune disease. And as a physician, I remember treating most of those patients with steroids or drugs that depress the immune system, immunosuppressive drugs. So if you've got lupus, for example, you're getting treated with Plaquenil and steroids. And it caused me as a physician to kind of be like, well, I haven't seen an autoimmune patient in a while. What's the standard of care for lupus? And I saw that there was a drug approved called belimumab or Benlysta that was targeting B cells, which made sense to me because as a physician, you actually monitor autoantibody levels in the clinic, which is meaningful. It's not experimental. These are CLIA certified labs, and you monitor disease activity by the level of autoantibodies. And I thought, wow, there's a drug approved that hits the B cells through one of the two mechanisms that cause B cells to survive and mature, and that's BAFF, also known as BLyS, B lymphocyte stimulating factor. And so I was getting to know this drug called Benlysta, and it was approved in lupus. It had been launched as an IV drug, but then converted to sub-Q, and the revenues were starting to get to blockbuster status, to about $800 million in revenue back in 2019. It's well beyond $1 billion now. And we thought, that's interesting. That's a clinically validated mechanism, and it's now looking commercially validated, which doesn't always happen. You can have an approved drug with an interesting biologic target, but it doesn't become a blockbuster. That certainly happens in our industry. So having that insight about anti-BLyS or anti-BAFF had us, the team, look back and say, well, actually, the biology tells us that BAFF is one of two signals. There isn't one signal. There aren't three. There are two signals. And we actually, B cell biology has been well explored both in academics and industry. And we know that BAFF and APRIL are those circulating cytokines. That's not a cell surface receptor. They're circulating cytokines. We thought, man, there really should be a drug that inhibits both BAFF and APRIL. And so we literally went to PubMed and looked for a molecule that inhibits BAFF and APRIL, and we found our way to this molecule called atacicept. Now, atacicept is it's a biologic. It's a fusion protein. It takes the native TACI receptor and binds it to an inactivated Fc portion. That's a very kind of logical approach to creating a drug. Enbrel, for example, is a fusion protein made of a native receptor and an inactivated Fc portion. So it's a really kind of straight down the fairway shot for, how do you make a drug? And the question is, how did that do in clinical trials? And there's a drug that was invented in the early 2000s, and then from a California company called Zymogenetics. It was partnered with Merck KGaA, and they tried it in several autoimmune diseases that, as a developer, I would consider kind of a double black diamond approach. It's like, okay, we're going to go for rheumatoid arthritis, where standard of care is super good. We're going to do systemic lupus, where patients are heterogeneous, and in trials, the endpoints tend to be composite clinical scores. These are really challenging drug development areas. And we looked at that landscape and we said, we love the target BAFF-APRIL. We think all of the characteristics of the drug, the half-life, the general safety profile. There was an integrated safety analysis of 1,500 patients. All looked like the safety profile looked similar to placebo. And yet, clear efficacy had not been established in these challenging autoimmune diseases. And so the decision that we did to take this drug into IgA nephropathy was one that was really based on the strategy that, let's test the drug in a really clean, clear population, a definable and accessible patient population, patients with biopsy-proven IgA nephropathy who are at high risk of disease progression. Let's take this into a robust phase 2 trial and prove that the hypothesis of reducing the immune complexes that cause disease could actually have a positive impact on kidney function. And as a physician, you know that if you measure protein in the urine or the serum creatinine and calculate a GFR, this is what clinicians do to measure how the kidneys are doing. If the GFR is less than 15, you're on dialysis and you need a transplant. So they're really clear, objective lab measures of outcomes. And that strategy is what we lined up back in 2019, 2020. During the year of COVID, we successfully in-licensed the molecule, did our last private financing of about $80 million in October of 2020. And that gave us the molecule. And we started to build the company, build the team, and kick off a phase 2 trial that we've now been reading out in a fantastically positive way. We're on our way to phase 3. We're going to have a phase 3 readout before this summer. And assuming that's positive, we'll be filing a BLA by the end of the year and planning on commercializing this drug in IgA nephropathy in 2026. So it's been an incredible conversion of the early days where we started in an area that was also an area of high unmet need, but the science told us we didn't have a path, and we found our way to a really compelling story, and the phase 2 results are really beyond our wildest dreams.
And can you tell us a bit about those phase 2 results? And I know, like you said, that the phase 3 readout will be in the summer. So that's truly exciting just to hear about the whole development process and through commercialization. I think that's and then throughout it, what really stands out to me is that the science leads it all. So
100%. I like to say, I think in our industry, we're lucky that it's a science-driven industry. So, in academics, it's possible to publish something. That's often the end goal, but sometimes it's not reproducible. We know that. But in industry, you have to be sure that your results are real. No pharma company, no biotech company wants an unsafe product. That is a really bad outcome for everyone. And I think having that clear in your mind tells you then trust the science. And I think there's so many examples in our industry where that's really important. I think that's certainly been true for us. So, the preclinical science where we discovered that our platform wasn't ready for development was an important moment where science guided us to really pivot. In this case, we had phase 2 data that again, so first, it matters what kind of phase 2 trial you run. We ran a multinational, randomized, placebo-controlled dose range finding study. We ran a pharma-like phase 2 trial. That gives us a strong sense of what is the right dose. And when you're modulating a B cell to try to treat an autoimmune disease, to us, it's really important that you get it right. You get the Goldilocks approach, not too much, not too little.
Right.
You need enough B cell modulation so that you turn down that overactivity of the immune system, but not so much that you're depressing the immune system. And actually, in phase 2, we showed again the safety between atacicept and placebo was really similar. And that was even in the context of the COVID-19 outbreak. So can you imagine being a patient with an autoimmune disease and taking a medicine, experimental medicine that messes with your immune system? It's a scary idea, but we certainly we enrolled the trial well, and patients who are on atacicept had no higher rate of COVID infectivity, no severe COVID, no deaths due to COVID. So it appears that we have that balance correct. So number one is safety, and the safety results over a 2-year period have been very reassuring. We, of course, want to see that validated in a phase 3 trial result as well. The efficacy was literally confirming of our hypothesis in a way that really was outstanding. So Atacicept inhibits BAFF and APRIL. BAFF and APRIL are elevated in patients with IgAN, so they overstimulate the B cell compartment. B cells and plasma cells produce a higher level of autoantigen and autoantibody. So galactose deficient IgA1 is the presumptive autoantigen that causes IgAN, and then there are autoantibodies of both IgA and IgG subtypes that are identified that are specific to the hinge region of galactose deficient IgA1. So this is a very well-known pathophysiology, and we dial all of that down. So the immune complexes that circulate is a really, you got these clusters of antibodies and antigens. They lodge into the kidney, which is our body's filter. You got these beautiful little glomeruli made of cells, and the glomeruli get inflamed, they fibrose, and they fail. And we're all born with about 1 million nephrons per kidney, so 2 million nephrons, and your GFR doesn't start declining until you lose half of those. So the patients we enrolled in this trial, on average, they're 35 years old. They are on an ACE inhibitor and ARB. They also had, kind of maximum supportive care, and yet they're still producing over 1 gram of protein in their urine a day. So these are patients who are going to lose their kidneys before the age of 50. They lose about 10% of their GFR annually. And the placebo arm of our trial in phase 2 showed that trajectory. So we enrolled the right patients, they're sick, and they're on dialysis before 50. So that's a terrible outcome. I'm 51, I've got 3 kids, I got a busy job, I've got a family to support. And man, if I were hooked up to a machine 3 times a week, that's a big impact. So you can measure it in suffering, morbidity. You can measure it in mortality. You're on dialysis, and essentially your mortality is similar to having stage 4 lung cancer or colon cancer. It's a really bad outcome. So patients on atacicept ended up having normal GFR at 9 months, at a year, at 1.5 years. And this past fall, we showed that effect lasted for 2 years. So the slope of GFR in these patients in phase 2 is minus 0.6, which is pretty close to -1, which is what patients without disease experience over their lifetime. So these are really seen as transformative data. And based on those data, we've really doubled down as a company, tripled, quadrupled down. We went quickly into phase 3. And as I said, we'll be reading out those results before the summer. And we're also starting to take atacicept into adjacent populations, patients with IgAN who may not have qualified for a phase 3 trial, or patients with other autoimmune kidney diseases, for example, like membranous nephropathy. There are certain forms of focal segmental glomerulosclerosis, or FSGS, and minimal change disease, in which patients actually have autoantibodies that drive that final common pathway. So our clinical and medical leadership within the company, our CMO, Rob Brenner, and his team have advanced atacicept into adjacent trials. We've gone really from about 2 clinical trials 1.5 years ago to over 7 clinical trials where we're now expanding our operations. So that's what you get when you've got a hypothesis that looks good, safety looks good. The initial disease where we're taking atacicept is an incredibly important unmet need. We hope to make a massive impact for these patients. We hope to create a very successful commercial launch. We're well on our way to doing that, but we're also expanding at the same time to make sure that we're maximizing the opportunity we've generated. So a really exciting time for the company. And we just spent last week as a full company, we gathered the US and global nephrology leadership to discuss, share our plans, and get feedback from our academic advisors. So we are, firing on all cylinders here at Vera.
Indeed. That's so wonderful and fantastic to hear. And just in terms of really hearing about how this therapeutic is really addressing such a significant unmet need in terms of these patients who are on the verge or inevitably will be on dialysis. And so you're able to essentially halt the progression of the disease, which is an incredible step towards, trying to treat this very serious disease. And so it's so fantastic to hear about. And in terms of, I think you touched on this in terms of selecting really the right patients. And then we hear a lot about patient centricity in clinical trials and patient-centric trials. And I always kind of question, what does that really mean? And so at Vera and for yourself, leading Vera Therapeutics, how does the company really instill that kind of a model? From what I hear, I can tell that is definitely there, but how does that play out in your clinical trials?
Yeah, I think the root of it is that I'm a physician, and the reason I'm not taking care of patients is because I think I can give patients a new medicine and provide physicians with a new tool. We do talk about patient centricity, but I'd also add physicians as one of our key stakeholders, our key customers, and it's something that I've experienced personally. When you face a patient in a room and you say, what brings you in today? And you diagnose something, the next thing is, what can I do for this patient? And it is a terribly uncomfortable position to not offer a patient a real option that helps them. So we really think both of patient centricity as well as, look, we're trying to serve physicians. Physicians are doing the great work of showing up and saying, what brings you in today? It's an ancient calling, and so we respect that as well. Last week, we had a patient come in to talk to the whole company. We had a patient in the room when we discussed Vera and our plans in the nephrology field with advisors over the weekend. So just in the last 7 days, we had two separate patients. So I think patient centricity needs to come from the top, from the leadership. The patient voice needs to be at the table. I think that it's great to motivate the whole team, whether it's the clinical team or the finance team, to hear about why we're doing what we're doing. So that's one of the roles of a CEO is to make sure that you're motivating everyone, reminding people of the mission and the purpose of the company. So that's a major thing that we do. We do it at least once a quarter. We bring in a patient to talk to the full company, and then making sure that we've got a patient's voice in the room when we're making some key decisions. So I think patient centricity is important. It's when you talk about getting a drug to market, there's a financial argument for that, but there's really a patient argument for that. Patients are waiting. So when we read out our phase 3 results before the summer, and we start the clock to our BLA filing and putting together something like 2 million pages to submit to FDA, we're counting those days, and we're thinking about every day that is later than it could be is a day that the patients who are waiting don't have access to atacicept. So that's how we think. That's how we act.
Excellent. And that's such a great point in terms of, physicians being an important stakeholder. And I actually heard the same thing from another CEO like yourself from a biotech just last week. And he said the exact same thing, echoed the exact same message that, physicians are an important player in this because they are also, they're like you said, trying to offer patients the therapies and treatments that work. And so that really does take a toll on them in terms of if there aren't any good therapy or therapeutic options. And so that's so important to highlight. And I think we'll probably be hearing more and more about that. So thank you for highlighting that as well.
Of course.
And in terms of, just the drug development landscape currently, what really are the current challenges in this ever-evolving space, laden with and filled with innovation and just so many happenings, and in terms of trying to bring a novel therapeutic to the market for especially complex diseases like IgAN. And so what are some of the challenges that you've had to navigate? And what have been some things maybe that have been easier now than, say, 10 years ago? So both challenges and the ease, I guess, or things that have been made easier now? If any.
Happy to start kind of with a broad view and then cone in on our situation. I think the elephant in the room today is that the macro, the geopolitical environment that we operate within is challenging. So there's no doubt in my mind that when there's peace and prosperity and collaboration, science and medicine flourish. And so as we have conflict and lack of agreement, that becomes a challenge. So, as we look back over the decades, collaboration between the United States and Europe, Asia, these have been incredibly important because you combine the resources of all of these great places. And so as those relationships cool, it creates real challenges. I also think that it's important to acknowledge with the concern that the United States may not invest in the biomedical research project, for example, whether it's funding treatments ex-US or, more importantly, funding the National Institute of Health is really foundational to what we can do as an industry. So nobody in pharma or biotech would argue that we don't need the NIH to do discovery-based, curiosity-driven research. I've got a teenager who's interested in biology and medicine, and I hope he can look to a future where he could be an NIH-funded investigator. And to disturb that that apparatus, that flywheel of innovation in this country and outside is, in my view, a real mistake. So that I keep in mind. That's a challenge. It doesn't affect us right now, but it affects us long term, and we're always thinking long term. And then I would say that the markets have been challenging. We have a bear market. So we went public back in May of 21. It's now 4 years later. Everyone always says, can't see around the corner, but let's just say it's remained a bear market. We've navigated effectively. Thank goodness that the science has translated into positive phase 2 results, and Vera has been successful in that market, but that hasn't been true across the board. And I think when results or situations are more ambiguous than ours, opportunities evaporate that actually have real medical promise. So those are some of the challenges that we face and have faced since our recent history as a public company over the last 4 years. I'll say that, I do think that it was wise to go public back in 04. So I mentioned that we in-licensed atacicept in about October of 2020. And then we went public less than a year later, maybe 9 months later. And why do we do that? We even were advised at one point that, market's not looking great. Maybe you should stay private. I'm thrilled that we went public. As a public company, as you get to late stage clinical development commercialization, first, it's important to be part of the conversation, and it's easier to do that as a public company than a private company. But really, it's about access to capital. So even in a bear market, access to cheap capital, more substantial capital is critical. These are wild benchmarks, but it takes about 10 years and $1 billion to develop a drug. And that's very challenging to do in a private market for both the quantity of capital, but also that timeline and the ability to evolve governance in the way that you need to serve the development program and evolve your ownership. As I like to say, you're talking to a founder. So at one point, I owned 100% of the company, which had nothing. And now I own a much smaller percentage of something that's worth something. And so our ownership has really developed in a pretty standard way, standard according to biotech, where ownership is diversified. We have an excellent board that represents the shareholders, and that evolution, I think, has made a lot of sense for the ultimate mission of the company to create innovative medicines for patients in need, to support the focus of Vera, which is in the development and commercialization of those medicines, and to support really an evolving opportunity that thankfully we've been able to earn over the past bunch of years.
And in terms of, I guess, innovations in biotech, we hear a lot about that. It's an innovation-driven field. And so what are some of the, I guess, current innovations that you're excited about and that are helping drive something like your lead candidate? And also, what do you see in the next couple of years? What are some of the, I guess, the trends, quote unquote, in the field and, up and coming technologies? We keep hearing about, AI and all of that. Are these just, buzzwords, or people just using them for the sake of using them? Or, how do you really apply these hot new technologies that everyone just wants to use? But are we using them correctly?
I think these are incredibly important technologies, but I think the first perspective I would offer is that every business needs to exist for a purpose, to solve a problem, and you have to be clear on what that is. You can pivot, but Vera has been focused on serving patients' human health. So the problem we're solving is medical. And the solution is most directly scientific. It's a drug, it's a product, and that product I like to kind of bookend new medicines from the simplest form of small molecules like an antibiotic, all the way to cell and gene therapy, a much more complex, hard to control, hard to reproduce. Cell therapy, gene therapy, oncolytic viruses, what have you. And then the middle is really biologics, monoclonal antibodies or fusion proteins. So thinking about that range, I'm so excited about the more complex end of the range. We've gotten small molecules down. You can genericize those. Monoclonal antibodies and fusion proteins are now reproducible. It's possible to create a supply, to create a business with a good margin around a fusion protein like atacicept, although it's hard to reproduce. So when we look at the potential for others to say, just copy what we do, we think that potential is extremely low. But the regulatory risk, the clinical supply risk has really been diminished over the last several decades. So that really is a sweet spot for us. And then as you move to cell and gene therapy, I mean, how can you not be excited about a cell therapy that can cure brain mets in a 30-year-old woman with lymphoma? I mean, I remember reading that article in the New England Journal about, 10 years ago and thinking, oh my God, I can't believe these results. So, the promise of cure with cell and gene therapy, it has to get solved with manufacturing, standardization, reproducibility, and then our payer system needs to evolve to a system that can support a cure. Is it $1 million a cure? Is it $500,000 per cure? I mean, we've got to figure out the economics of that because the human health benefit is massive. So I would just sort of bookend that piece. AI and other things I'm super bullish about. So we talk about AI in every aspect of our business. To me, the most exciting thing is that DeepMind took on the idea to predict the protein folding of all known proteins in the mammalian genome, and they did it in like 2 years. I mean, that is, yes, it needs to get validated in the wet lab, but I mean, that is just the coolest thing ever. If you did any biochemistry and you know about primary structure, a string of amino acids, the secondary structure of alpha helices and beta pleated sheets, and then tertiary structure, how they fold, and quaternary structure. And then you think about that and you're like, of course a computer can solve that. And then they did. I mean, how can you not be excited? That's totally awesome. And then on the flip side, we think about how is this going to change contract review on the legal side? How is this going to influence time from phase 3 data to FDA filing? because AI can help you write your BLA or your NDA. So, those aspects in the business, I think, are incredibly exciting and ripe for innovation. And we are, of course, thinking about all those things.
Mm-hmm. Yeah, that is indeed very cool. And we talking about the protein structure and taking me back to biochemistry second year. And yeah, like, I mean, first it was the human genome, and now, we're talking about just elucidating the structures of all of these proteins. It's truly incredible what technology has enabled us to do, and really exciting to see what the future holds for sure. And we'd like to know what the future holds for Vera. I know you mentioned that there are a couple of very important milestones coming up this year. And in addition to that, what really are the short and longer term goals, which you mentioned that, you have very clear long-term goals at Vera. And so we'd love to hear about that.
Yeah, the short-term goal is, of course, to file our BLA. We ran a very phase 3-like phase 2 trial, so we have high confidence that the phase 3 result will reproduce the phase 2 result. So it really is an unlock for us as we move into BLA filing this year. So that's really the short term, and then the other near term, getting into 2026, is successful commercialization. And I see atacicept as a transformative new therapy. We want to make sure that the data and the opportunity is well understood by the physicians who we hope will prescribe this in the future. And so we're very focused on leading this innovation in the nephrology space. So my background in infectious disease and HIV medicine, HIV drug development provides an important template for what can happen in a disease area. And you need to communicate. One thing I love to say is, sometimes people in science or medicine say, oh, the data speak for themselves. And at Vera, we're like, no. Data do not speak for themselves. Data need to be spoken for. They need to be communicated in 1,000 different ways to all the different stakeholders, to regulators like FDA, to payers, to physicians, to across the board. And so that's a near-term goal for us, is to effectively communicate the value story and the urgency to treat these patients and the window of opportunity that exists for us to innovate in the nephrology space, the autoimmune nephrology space. So that's really the near term for Vera. Longer term, as a business leader, I am thrilled to get to a point where we will have sales revenue and start to be a profitable company. That's going to be a very satisfying moment for us, and I'm looking forward to getting the company to that stage in the near term. I think there's a real opportunity to develop and build a company that is the biotech company of the future. We're earning that opportunity every day.
I love that. And Dr. Fordyce, thank you so much for all that you do in leading such wonderful, fantastic, and such important and significant work in the field for a disease like IgAN and the field in general in renal diseases. So thank you so much once again for your time and for all of the work that you do. And we'll definitely be keeping an eye out on Vera at Xtalks to keep updated on all of the milestones and updates that will be coming from the company soon. So we look forward to that and wish you the best of luck.
Great. Thanks so much for the chance to tell our story today.
Thank you. That's the end of this episode of the Xtalks Life Science Podcast. If you liked today's show, don't forget to rate, review, and subscribe. Thank you, and we will see you next week. Bye for now.
To join the Xtalks community, visit xtalks.com and register for exclusive access to curated content like live webinars, news articles, and educational videos. The views and opinions expressed in this podcast are those of the speakers sharing them and should not be taken as professional advice, nor do they necessarily reflect the policy or position of Honeycomb Worldwide. For further information, email us at podcast@xtalks.com.